Hi everybody. This is a lesson, a pre-lab lesson for our lab that will be tomorrow. In tomorrow's lab, we're going to be extracting phosphorus out of plant food using a process called gravimetric analysis. And what I'd like you to do, what I would like to do today is just kind of walk you through the procedure, make a couple and the calculations, so that tomorrow when you come in, we can just get right down to business with the lab. The filtration takes a while, so while we're filtering, I think Bryce's group and Evan's group and Sarah's group, have to present their airbag stuff to us. So while we're filtering, we'll do the airbag stuff. So that way we can get right down to business and right to work. So if you would take out a sheet of paper, that'd be great. I'll kind of walk you through the procedure as to what we're doing and walk you through the calculations and how you get your calculations. And then tomorrow you could just get right down to work. So in our lab, basically you're going to be starting with plant food. This is my box of plant food. And plant food has in it a lot of nutrients that plants need. Nitrogen, potassium, and phosphorus. And on the box of plant food, it always gives the percentage phosphorus. And so our job in the lab will be to calculate the percent phosphorus. The way we're going to get the percent phosphorus is by precipitating the phosphorus out and weighing it. So the first thing we're going to do is dissolve the plant food in some water. We're going to add some distilled water to the plant food, and we're going to add about 20 mils. So you're going to weigh out about 1 gram of plant food and put it in a beaker. Okay, so pretend this is our beaker. And then to that, you're going to add 20 mils of distilled water. And you're going to stir it. You're going to stir it in an attempt to dissolve the plant food as much as you can. And it will dissolve. Remember, this is not soil, this is plant food. You're then going to filter it. Okay? So you're going to filter your mixture. And then you're going to add another 10 mils to this beaker, to your plant food. So after you filter it once, this is your filtrate down here. That's what you're going to keep. You'll wash your original beaker with another 10 mils of distilled water. And collect your filtrate. What's on the filter, anything that did not dissolve here, you can go ahead and discard that. Okay? Because our dissolved ions, our phosphorus, our potassium, our nitrogen, they'll all be in the filtrate. That's what we want to keep. Okay? While someone in the group is doing that, another partner can get ready the other reagent that we're going to add to the filtrate. And that is going to be some Epsom salts that are dissolved. We want about a 10% solution of Epsom salts. Epsom salts are just magnesium sulfate. Okay? And so to make that, you're going to weigh out 2.5 grams of magnesium sulfate. And you're going to add it to 25 mils of water. Okay, you're going to stir that, and that will be your solution of magnesium sulfate. Okay? Then you're going to take this, once you have it dissolved, and you're going to pour it into your filtrate. And that will begin the precipitation of the phosphorus. And then you're going to add about 40 mils of ammonia to that beaker. And you should see a white precipitate form. That should make a white precipitate. Oh, that's out of the... That will make a white precipitate that you're then going to set up more filtration for. So in this filtration, you'll weigh filter paper. Because this is the filter, this is the precipitate we're going to collect. And you'll put your filter paper in your funnel, and then you'll filter again. And this time we want to collect the solid. And this is what takes about 45 minutes to gravity filter. So we're going to collect the solid in the top of the paper here, the top of the filter paper. And we'll let that dry over Thanksgiving holiday, and then when we come back, we will weigh it, okay? So by weighing it, the name of our, I'll tell you, this is a crazy precipitate here. The precipitate is actually this crazy thing, MgNH4PO4.6H2O. It's a hydrate that has water as part of its structure, but what we're really interested in is the P. So after Thanksgiving, you'll come back and you'll get the mass of your precipitate and your filter paper together. Okay, you'll weigh both of those on Monday, and then you can get the mass of precipitate you made by subtraction, right? So that would be the mass of this one, the mass of precipitate and paper, minus the mass of the paper that you weighed before you filtered it. And that'll give you the mass of precipitate. From the mass of precipitate, you can get the moles of the whole compound of precipitate, because they actually give you the formula mass in the lab. The formula mass is 254 grams per mole. If you have the moles of the precipitate, you can get the moles of nitrogen, or excuse me, we don't want nitrogen, we want phosphorus. The moles of phosphorus inside that, because in every 1 mole of the compound, or there's 1 mole of P in every 1 mole of the whole compound. So that'll give us the moles of phosphorus, and if you have the moles of phosphorus, you can get the grams of phosphorus, right? Simply by multiplying by the formula mass of phosphorus. So the whole point of doing this is to actually calculate the grams of phosphorus that we had, and then we can get a percent phosphorus by taking the grams of phosphorus and putting it over the grams of plant food that we originally weighed, which should be about 1, right? It was about 1 gram, and then multiplying it by 100. And since the plant food actually gives us the percent phosphorus on the label, we can calculate our percent error. So you're going to be performing gravimetric analysis, which is an important process in science. For example, if you wanted to know the amount of iron in a raisin, you could precipitate out the iron, get the percentage in a raisin, and then you would know how many raisins you needed to eat in order to meet your daily amount of iron. So it's an important process that we use in a lot of different areas in science, and you'll be performing it by determining the percent phosphorus in plant food. So that's it. Come prepared tomorrow. I'll give you the lab handout. Bring your kind of roadmap here with you. Hopefully you made sense of it. If you have to rewind me a couple times, do so. And we'll see you tomorrow. Have a great night, everybody. Bye-bye.